Okay, good morning, everyone. Thank you so much for being here. My name is Will Zeiger. I'm one of the movement disorders neurologists here at UCLA, and we're really excited to have you here on what it turns out is World Parkinson's Day. I did not know this when we were scheduling this symposium, but today is World Parkinson's Day. So by being here, I think we are all taking a small step towards the goal of this day, which is to try to end Parkinson's disease. We have a really exciting and interesting morning set up ahead of us. Today's symposium is a little bit unique in the sense that you're going to be hearing directly about research from the researchers that are doing it. So you're going to be really hearing about the cutting edge of what we know about what causes Parkinson's disease and how we think about translating these scientific insights from the lab into the clinic, ultimately to better diagnose and treat Parkinson's disease. So we'll start off kind of on the more basic end of that spectrum. Dr. Bronstein will give us an introduction about what causes Parkinson's disease. Then we're going to move on to Dr. Peng, who's going to talk to us about efforts in his lab to develop novel therapies to treat Parkinson's disease. And then we're going to hear from Dr. DiBiase, who's going to tell us about a really unique type of cell in the brain called microglia and what their potential role is in how Parkinson's progresses. At that point, we'll have a break and we'll come back after the break to hear 3 more talks, which I'll introduce at that time. So before we jump into the research, I just wanted to go over a little bit of terminology so that we're all on the same page. We're going to be talking about a lot of scientific concepts today. And in particular, we're going to be talking a lot about the brain. So this here is a picture of the brain. If you sort of took a view of the brain from the side, that's what we're looking at here. And within the brain, there's a number of different types of cells that make up the tissue of the brain. A lot of times we focus on these brain cells here, which are called neurons. These are the electrically active cells in the brain that send signals, helping us to move and sense and think. But these cells can't exist without a bunch of other cells in the brain, which include cells like oligodendrocytes that provide this insulation around those neurons, astrocytes, which also help to support signaling and health of these brain cells, and then microglia, which I alluded to before, and Dr. DiBiase will tell you all about their role in the brain. There are many parts to the brain, but one of the parts that you're going to hear about today is in this part of the brain here. And if we were to sort of take a section out of that part of the brain and look directly at it, this is what we would see. And if you look closely here, you can see this black part here, which is called the substantia nigra. And in this part of the brain are a number of neurons that produce a substance called dopamine, which hopefully you all have heard about. And we think this is especially important in Parkinson's because if we look at the brain of someone with Parkinson's, this black substance you can see is greatly reduced. And that's because those brain cells that produce dopamine end up degenerating or dying over time. And now dopamine is important for a number of things, and it sends these dopamine signals throughout different parts of the brain to help us do things like move, make decisions, regulate emotions, and a number of other aspects of Parkinson's disease that you may hear about today. Another term that you may hear about is this term alpha-synuclein. So alpha-synuclein is a protein. Cells are made up of proteins and other molecules. And alpha-synuclein has this tendency that it can get clumped up and form what we call aggregates. And you can see pictures of these aggregates. These are these dark brown spots sitting inside a neuron. And these, when they clump up together, we call Lewy bodies. And alpha-synuclein is the major component of these Lewy bodies. And as you'll hear about later today, we think that this alpha-synuclein can cause all sorts of problems for brain cells. And I'm not going to go into the details here because you'll hear about that much more in later talks.
Excuse me, doctor.
Yeah.
Do you know if that microphone is working?
The microphone up here? I think it's working. Can you hear what? Okay. It's a little low. Let me see if I can turn it up here. Maybe just have to stand right here, huh? Yeah. There's the hand mics here too, if people want. Yeah. Okay. So we'll try to stand a little closer to the microphone. Thank you for pointing that out. The last thing that I want to point out here is that today you're just going to be hearing about a really small sliver of the amazing research on Parkinson's that's being done at UCLA. So this is probably not a comprehensive slide of all the people who are involved in Parkinson's research in one way or another at UCLA. And so we're just going to be hearing stories from 6 researchers, but just to give you a sense that there's a lot more going on than we could possibly cover in one morning. So with that, I'm going to turn it over to Dr. Bronstein, and he's going to tell us about what causes Parkinson's disease. All right, thanks.
Thanks, Will. So I want to thank everybody for coming early on a Saturday morning. It shows a commitment. We really appreciate the support. And I want to thank Dr. Zeiger, who put this together. And just a little background. So Will applied to get what's called an affinity group at UCLA through the Brain Research Institute, and was able to get a grant basically from them to help organize people that are interested in one topic, and in this case, Parkinson's. So he's brought together people from all different departments, and it's really helped bring us together. So this is a representation from that affinity group, and thank Will for the affinity group and for today. So I was just sad to see on the program that he's not speaking. He does some very cool work with high-powered microscopes and stuff. So hopefully you'll get to see his work someday. Anyway, so I'm going to talk a little bit about what causes Parkinson's and what we think is going on. You're going to see some recurring themes, which is good because it helps you remember, at least for me. You know, you learn everything once and then you see it over and over again and you kind of store those memories. So you'll hear things over and over again, which is a good thing. So how do we, oh, okay. All right. So you always have to have the introduction slide. There's a lot of people with Parkinson's, somewhere about 1 million, about 1 in 100 chance for most people. Women don't get it quite as much as men for reasons we're not sure. Peak age is around 62, but the older you get, the higher the risk. It's a, as you know, big burden to people and society. So finding treatments and cures is a pretty high priority. Oh, all right. So you've already seen this picture. So you know these things are important when you see them over and over again. So this is my version of that. So let me see if I can. So this is the area of the brain we're talking about. And if you cut it this way, you can see this is the substantia nigra, nice and dark. And this is Parkinson's one, where those cells are being lost. Now, when I started doing this research, very beginning of my career, this is about all we knew. So this is a section in the brain, and this is stained, non-specific stain. And this abnormal structure, the Lewy body that Dr. Zeiger mentioned, this is the pigment in there that makes it black. So that's what tells us that this is a dopamine cell in this brain tissue. These are astrocytes and all those other cells that he mentioned. But this was abnormal. We didn't know when I started, we knew nothing about it other than it was there. And we didn't know what was in it. And so we've come a really long way in the last 30 years. So alpha-synuclein was first identified because there was a very rare family that had a mutation. They found out where that mutation was, turned out to be in this gene called alpha-synuclein. They made antibodies, and those antibodies bind specifically to a very specific protein. And sure enough, it's brown now because that antibody is staining it. And as Will said, it's the major component of this. So this has really been like biggest finding, one of the biggest findings in the last 30 years. And it turns out that it stained the Lewy bodies in that family, but it also stained it in people that didn't have a genetic form. So it turns out that most of us believe that alpha-synuclein is really central to Parkinson's and almost defines it in many people's minds. So you're going to see a couple different versions, and Dr. Peng is going to talk a little bit more detail. He's a real expert in this. So for somebody more of a novice like myself, I just think of it as normally this protein is kind of unfolded, unstructured, and then it starts folding in an abnormal way. And there's many reasons why that may misfold, which is one of the things that we study. Why does it misfold? Because when it does, they start clumping up and they're toxic. It's still not known whether these one form of these clumps are called fibrils that are really resistant to almost anything. And that's what the heart of the Lewy body is. And we don't know if that is really a way to protect the brain or actually it's toxic. Most of us believe that these things called oligomers, that intermediate clump, is the toxic form, but it's still not 100% agreed upon. So this concept of templating, it's kind of like, anybody remember those crystal seeds when we were kids, and you get a little bit of a start and all of a sudden it grows. And that's what really happens with alpha-synuclein. It's like a seed. So this is the abnormal folded one, and that will convert a normally folded one into abnormal ones, and that clumps up. And when they clump up, they're not only toxic to the nerve cell, but really importantly is that they actually transmit that clump to a neighboring neuron and starts the process in that neuron. So we call that transmission or spread. So this happens very slowly. It happens over decades in people. So it's a very, very slow process, which means it's an opportunity to interfere with that. So for a variety of reasons, we believe that it actually starts either in the gut or in the olfactory bulb. Maybe see it down here. Those are the earliest places that we see these Lewy bodies, the abnormal forms of alpha-synuclein. And then it spreads up this nerve called the vagus nerve, and then gets into the central nervous system, the brain, and then it can spread throughout the brain. Again, this process we think could be starting 2 decades before the first symptoms occur. So very important concept. We're not sure that everybody starts this way, but we think the majority of people do. So this is kind of a timeline of when things happen. So when you have the first symptom, that's times 0. So it turns out when we do these epidemiology studies that people 20 years before that started reporting reduced number of bowel movements, constipation starts. People maybe start getting up more at night to go to the bathroom. Maybe they don't smell quite as well. Those are the earliest symptoms of Parkinson's disease. It's just not everybody who has constipation is going to get Parkinson's disease, but most people who get Parkinson's get constipation. It starts years before. And people start maybe acting out their dreams a little bit, start having a little more anxiety or depression that's unusual for somebody. That's not normal. And then all of a sudden, eventually, you start getting a little bit of a tremor. And then it can progress from there. So all of these symptoms actually have places in the nervous system that account for that. That's why we think it spreads, because we see these symptoms occurring over time. All right, so this is really the key question that my group and a lot of people here at UCLA study is what starts this process and makes it continue? Why do some people progress faster than other people? So the short answer is you have this fertile genetic ground. So we all have a genetic background, and environmental factors alter your risk. So that's what we study, is what are the genetics that make you fertile, And what are the environmental influences, especially those, because those are modifiable. Those things we can change. Can't choose your parents, but you can change what we're exposed to. So the genetics of Parkinson's has really been very interesting. When I started, nobody thought there was a genetic influence at all to it. And then that rare family was found with the alpha-synuclein, so people started looking more. And as the tools of genetics grew, they started identifying more and more genes that can cause Parkinson's. There's what's called dominant and recessive, whether you need one copy or two copies. But there's several genes that do it. This is a short list of it, but all of them added together may add up to maybe 5% of all Parkinson's. So many genes can cause it, but they're very rare. But what they do is give us insight into how you can get Parkinson's. So we use that as a model to say, well, this gene is like the garbage disposal for protein. So let's look for things that alter that, because that is a pathway the genes use. So maybe other things use that as well. So we've learned a lot from these rare genetic causes, even though it does not cause the majority of Parkinson's, it's the minority. But then there's a little bit more of a complicated story. So we all have variations in our genes. They're called polymorphisms. And it turns out that some polymorphisms are a little bit more common with people with Parkinson's than other people. So we think that some of these genes confer a little bit of risk. And there's many of them. You can see there's over 30 of them now, that even more depending on which population and different ethnic backgrounds or different ones. But all of these confer a little bit of risk. And if you add them all up, and along with the other genes that I showed, it's about 30% or so. Genetics contributes to Parkinson's disease. So that's the fertile ground we're talking about, is all of these put together. That's the majority of people with Parkinson's have a little bit of increased risk due to many different variations. Again, they're not mutations, they're polymorphisms or variations. So if you put all those together, this is a cartoon that, of the different ways that I think that people can get Parkinson's. So this is synuclein here. And then for reasons that we're going to talk about a little bit, it can get clumped up and form these proteins, and that eventually leads to cell death. Inflammation, you're going to hear about microglia in a bit with Dr. Biase, and she studies this period. And so there's even a number of trials now to see if cutting this inflammation reduces this amount of cell death. There's also the energy center of the cell called mitochondria. And there's evidence that mitochondria, if they're disrupted, form a lot of the free radicals or oxidative stress, and that seems to contribute as well. This is what I'm going to focus on now, which we've been studying a lot. This is a process that helps break down those clumps. So it's like a garbage disposal. So if you can't break them down as well, this will accumulate and favor this process going to the oligomers in depth. So this is one of the things we study, are these protein degradation pathways. All right. So we talk about associations a lot. So that means we take a big group of people, and some have Parkinson's and some don't, and we say, what's different about those? So people with Parkinson's tend to be older. So the older you are, the higher the risk. Males tend to get it more often than females. Turns out that the biggest association is smoking cigarettes. People who smoke cigarettes have a much reduced risk of getting Parkinson's disease. So we're going to talk about that in a second. That doesn't mean it's protective, and don't go out and smoke. It turns out drinking alcohol and drinking coffee are associated the same way. But what we're going to talk about in a second is associations don't necessarily mean causality. I'll give you an example of that in a minute. Head trauma increases your risk of Parkinson's. Exercise reduces your risk of Parkinson's. Mediterranean or Main diet, maybe a vegetarian diet, reduces your risk of Parkinson's disease. Exposures to pesticides increases your risk. Air pollution increases your risk. A bunch of solvents increase your risk. So these are all associations. And so what our job is to figure out what these are and how they do it and see if they're actually causing it. So this is an example. There's so many of these examples. You can look up association versus causality. So if you look at people who eat, when people eat ice cream over the course, they eat it, mostly in July, and you look at drowning deaths and they associate with it. So if you look at drowning deaths, they'll say, oh, those people eat more ice cream. Does eating ice cream obviously doesn't cause people to drown. It's just it's an association, but it doesn't cause it. So there's many things like that. People with lung cancer drink more, have a higher rate of drinking alcohol, but it's not because alcohol causes lung cancer, is they tend to be smokers or around smokers more, and it's the tobacco. So all the time you hear in the news a new thing causes it. They're just showing associations. And the majority of those associations, honestly, aren't causal. And that's our job, is to try to figure out what if those associations cause it or not. And I'll talk a little bit about how we do that. So why do we study the environment, or at least I do, and people I work with? One big reason is 70% is unknown. What's causing that other 70% of Parkinson's disease? Now, if you think about, I said the pathology starts in the gut and in the olfactory bulb. That's how we interact with the environment, right? We inhale things and we eat things. So it's a clue that maybe it's the environment helps trigger this process. Okay. It's really hard to study the environment because the exposures presumably are happening 20, 30 years before you get the symptoms, right? You talk about it starts, we don't know when you have constipation that you probably are getting Parkinson's. So what may have triggered yours, or some of the factors were probably like 30 years before. So we try to go back and find those things. Those places are gone, or it's very different sources of water and air and all that stuff. So that's one reason. It's not so easy to diagnose people right early on. So many of the studies, people say they have Parkinson's when they actually don't. They have some other cause of the same symptoms. And it's really kind of tough to model in animals. We can't, nobody will give us a grant to study something that takes 40 years to happen. So we have to try to compress it into a short amount of time. So what we do is we don't really model the disease. We model aspects of the disease. So we do different models for different reasons. And I'll show you a couple of those now. So our approach is to help find these associations, and then once we find it, use animal models to try to determine if they actually cause it or not. Okay, so pesticides were one of the earliest things that were thought to be associated with Parkinson's disease. This is a map of pesticide use in California. This is the Central Valley. And it turns out in the Central Valley, about 20% of all the United States pesticides are sprayed there. There's a lot. And fortunately, California was smart, and 40, 50 years ago, they established what's called the pesticide use registry or report. And there, every commercially applied pesticide has to be cataloged, what they're spraying and when, because when you say pesticides, it's hundreds of different, I got 5 minutes, so I'm going to start speaking faster. Hundreds of different chemicals, and we have to figure out which ones are doing it. And so Beata and Ritz is one of the collaborators of mine for 25, 30 years. And she came up with a way with what's called geocoding, so that she can match your address from 20, 30, 40 years ago and estimate what you've been exposed to, because we know what was sprayed and where. And this is the pesticides being sprayed. in the Central Valley. And I've been sprayed many times. I've been going out there for 25, 30 years. Kim Paul, trained under her, is now one of faculty, and she came up with this one example. So this is called chlorpyrifos, is a pesticide, still legal. And she found from this study, we call it PEG, Parkinson's Environment Gene Study, that it's about 2.5-fold increased risk if you've been exposed for about from 50 years ago. So people that in the central, happened. Oh, I got to go backwards. Okay, well, I'm just going to talk because so I don't have to go backwards. So what we did was, so we found that chlorpyrifos was associated with an increased risk of Parkinson's. So now we want to see, does it actually cause it? So Hassan, who's up here in the top, Dr. Hassan, Kazi Hassan, he led this project. And what we did was expose, you can see in this chambers down here, mice to aerosolized chlorpyrifos, just like the people in the fields are being exposed. The animals breathed it in, and then Dr. Zeiger and Dr. Peng helped analyze these mice, and they didn't move as well after they've been exposed for several weeks. We look inside their brains, and their brains had many of the aspects of what we see in Parkinson's disease. They lose dopamine cells, alpha-synuclein accumulates, and they form these aggregates, which you kind of see here, of abnormally misfolded alpha-synuclein. These are the microglia. These are the inflammatory cells in the brain. And you'll have to take my word for it that these look more inflamed than the ones in the control. So they get rounded, they lose a lot of these arms. Dr. Biasi will talk a little bit more about this. But so it has many of the characteristics of the brains we see with Parkinson's. This was a movie, I don't have time right now, but we use zebrafish as well, because zebrafish are transparent, they grow really fast. So this is a fertilized egg, and within 24 hours, it already takes the form of a fish. And then this is a 5-day-old one where we had transgenic, and they light up. These are dopamine cells lit up. So we can actually see them in living fish. So we created, it's looking for the videos, but I'm trying to move forward. How do I advance? Yeah, okay. So this is what the fish look like. They're clear and they have less dopamine neurons. I'm sorry, I keep hitting the wrong ones. All right. And there's less dopamine neurons, and they're only exposed to very, very low levels of chlorpyrifos. And they form some abnormal folded proteins as well. So again, has some of the characteristics, but the main reason we're using the zebrafish is because we have one that looks at that garbage disposal, that breaking down the protein. And it turns out this is an assay that Dr. Barnhill in my lab has developed, and shows that these little dots here, those are little packages being taken to be chewed up. And when chlorpyrifos is there, they don't chew it up very well. So we think that is the cause of how chlorpyrifos works, is by inhibiting this garbage disposal, allows this to accumulate and causes cell death. So to answer the question that we started with, what causes Parkinson's? It's a little bit of everything. It's the accumulation of multiple risk factors. Things like genetics is a risk factor, what you're born with, but many things. Pesticides, some people get a very heavy dose of chlorpyrifos, and that may be a major factor. Other people had a big head trauma, or other people have a couple really bad gene mutations. So everybody has a slightly different cause. They have their unique cause. That's why every Parkinson's patient is unique. And but fortunately, many of these things are modifiable. We can change. You can exercise. You can eat a good diet. You can avoid exposures. You can really take control. So even though you already have Parkinson's, we think many of these things overlap with progression. So, which is what we're studying now. So with that, I'm going to say a lot of people did the work. These are other people that contributed to all the work we're doing. This is Maricel. just got her PhD. Michael's an undergrad, and Sharon has a PhD who helps run the lab. So with that, I'm going to stop, and we're going to do questions at the end. Okay, thanks very much.
All right, so we're going to save questions till after our third presentation this morning. So you have a chance at that time to ask questions for our first three speakers. But now we're going to move on to Dr. Peng, who is going to tell us more about that protein alpha-synuclein and how we might design therapies to prevent it from clumping up. Thanks, Dr. Peng.
Yeah, good morning, everyone. I'm a basic scientist. So what that means is like in 90% of my time, I'm talking with other scientists. I think this is my first chance to really kind of facing the patients, which is so exciting and a unique opportunity for me to share our research. So as Dr. Zager has introduced, my lab is really focused on this kind of a drug discovery side. You guys have heard about this protein again and again this morning. So this protein is, let me change this a little bit. Yeah, so this protein called alpha-synuclein. So like if you want to name just one protein for Parkinson's disease, people will probably tell you this protein, just because as Dr. Zager and Dr. Bronstein has introduced, that there's genetic mutations there, and the patients, they all have this synuclein, very, very big synuclein aggregation in the neurons. So look at here, this is the neuron. Usually, if you stain for a protein, you could barely see it. It will be a diffused signal. But then look at how big this is. It's almost like the size of a nuclear. So that's the huge amount of protein just come together. So if you want to remember one thing, just to remember this is a bad guy. Okay, that we try to target. So what is even worse is that if you think about alpha-synuclein, your body didn't make it to make you have a disease, right? So it must have a physiological function. So in normal case, like in kind of, I would say mass majority of the cases, synuclein is normal. It has its role in the presynaptic terminals to transmit neuron signal. But in PD patients, these proteins just change their structure. They take a different shape to becoming this bad guy. But what's even worse is that this bad guy not just don't perform their function, they actually affect the good guy. They change the good synuclein structure to the bad structure. So that way, it kind of the bad guy amplify itself by keeping changing more of the good synuclein into the bad synuclein. And eventually, all these bad proteins, bad synuclein, clump together, form these very, very long fibers, and become this huge protein complex. That becomes the real issue. So that's a very bad process. And what is even worse is that they could travel. Okay, so even if all the things is limited to one cell, that's not that big of issue because our brain is smart. We kind of compensate ourselves. Even if we lose some part of the neurons, we still could function well. But the problem is that these bad guys, they drive and move from one cell to the next cell and repeat this amplification process in this recipient cell. So now if you think about that, you have this bad guy moving in the brain and it affects more and more cells, more and more brain regions. That's the reason why these symptoms get worse and worse every single year. So that's kind of the whole picture. What we are trying to do is trying to target every single step in the life of this bad synuclein in order to find the potential therapy to block its formation, to block its transmission, or block its amplification. So I will cover all three aspects. So this is kind of a very detailed view of this kind of bad synuclein structure. So it's essentially, you can see, this is actual isolated bad protein from the brain. And you can see it looks like a fiber, right? And you take a closer look, you can see it's multiple layers of synuclein all stacked together, form these huge long fiber structures. That's very toxic. So the first step we try to take is to target here. So we try to block this fiber from formation, basically disrupt the structure, okay, to try to recover it to normal conformation. The problem is that we have over like 10 to the 60 different small molecules if we only consider ones that has the potential to be a drug. How would you be able to find that which one is able to break this kind of very big fiber structure? So the approach taken in my lab is like we combine this AI-based drug design with a very powerful experimental validation platform to kind of try to identify the small molecule that target this particular structure. So this is kind of the first strategy we've taken for AI is that this is kind of a de novo design. If you imagine that it's like designing a molecule that never exists from scratch. The way we do that is you take the pocket. So this is a kind of just example protein, it's not synuclein, but each protein have something, it's like a hole in the ball, right? You try to fill the hole with a small molecule to disrupt the function of that ball. So this is that hole, and the AI model will randomly put some atoms there, and put more and more and more and more, and eventually he will randomly generate some sort of small molecules to fill up the space that could potentially target the structure. So this is many people using this right now, but the challenge is that this is what a normal regular protein looks like, right? It's essentially a ball, and you try to target this small pocket here. But look at synuclein. This bad synuclein is totally different thing. It's a very well-known fiber with basically some tunnels on the surface that is very different from the traditional drug target. So if you run the traditional AI model on this, what you get is essentially like this. The model is trying to put these very, very small molecules into this big tunnel. What happens is that it couldn't cover the tunnel and have no effect at all. So that's the challenge, and that's where we started. So we think for a molecule to efficiently targeting this kind of very known fiber structure, they have to have this repeated structure because the fiber is the synuclein fiber is actually many layers of synuclein stacking together. So it's a repeated structure. It's not a random structure. So if you want a molecule to target this huge structure, the molecule by itself have to be able to repeatedly, right, bind to the same pocket again and again. In other words, the molecule have to have some stacking properties to be land on top of each other. So that's the idea, and that's the kind of most essential improvement we made to this AI model is that we teach them, okay, this is not what you see before. This is very unique. We call it a kind of a repeat apartment theory that if you put one item in one place, I force you have to do the same thing in the next layer and the next layer. So that the AI now becomes smarter. You know, okay, I not only have to deal with one layer, I have to do this repeatedly for multiple layers so the molecule could stack in on top of each other. So this is a simple way to do, but we spend a lot of effort on this. And this is what happens after you achieve that. You can see now the molecules designed by the AI looks much better, that the molecules stacking on top of each other, and they kind of each layer interact with one layer of synuclein. So that is a so much more promising therapeutic region than what we get before. So that's essentially the first stretch. The second stretch is not really a design, it's kind of a learning process. You're basically just telling the AI, oh, this one looks good, that one looks bad, and you train it with thousands of compounds that include data. And then the AI will give you some feedback, and you test it and tell him, okay, you get all of this wrong, and you get some of them right, just do it again. So after multiple rounds of training, the AI will start to learn some pattern. Okay, this one looks good to me, so maybe I can kind of pick up those ones smarter. So this is essentially a screen process to help you to kind of search for the enormous chemical space for the ones that looks reasonable for you. So we combine both of the effort to give us candidates of compounds that could potentially be used to target the synuclein fibers. And the problem is that the AI machine generates so many molecules. That's a huge amount. How are you going to test them, right? So for that, you need a model. You need a very efficient way to mimic synuclein misfolding in your system. So this is what we did. So we purified synuclein protein. So this is not from patient, but rather is artificial synuclein. And then we can make synuclein form this fiber structure that you can, if you look at the picture, you can tell this looks very, very similar to what we see in patient. And then we culture these primary neurons. So these are neuron cells from mouse. And then we treat these neurons with these artificial fibers. And you can see they induce an enormous amount of misfolded synuclein in the neurons. So this gives you a very, very good model system that you can mimic this misfolded synuclein in a neuron cell. And what is more important is that we build this system to a very high throughput format. So instead of culture the cells one by one, we culture them in this 384 well plate. And we can treat all of them together and test hundreds of compounds in a single batch. So this is like, you don't test one compound at a time, you test hundreds of them all at once to see whether they could target this bad guy. So that actually dramatically improved our efficiency and also gives a very critical feedback to the AI. Like, okay, now you have the data. Like I tell you, this one we tested is bad and those ones we tested is good. So after so many rounds of optimization and combine these two very powerful tools together, we essentially get several very exciting compounds. So you can see this is the actual data. And you can look at the green signal here is staining of the neuron itself. And the red signal is the bad synuclein that I mentioned to you. And you can see this is without treatment. This is after you add those compounds into the cell. You can see how much red signal has been reduced. And we have not just one, but a couple. I only put 4 here, but we have more. You can see a very dramatic reduction of the pathological synuclein signal in the cell culture. So that really gives us hope that, okay, now we get something like demonstrate that this compound could reduce synuclein pathology. And the one thing that I want to emphasize is that this is not end, right? This is a continuous pipeline of AI combined experiment that we expect to get more and more potent compounds. So we have an option to choose which one we will move into clinical trials. So that's the first part. We are targeting this side. The second strategy is blocking the trafficking, right? So even if we kind of couldn't target this, if we can block synuclein goes from one cell to next cell, we could still prevent the disease from getting worse and worse every single year. That is still a very promising therapeutic strategy. So the question is how? Because the transmission mechanism is super complicated. How do you know which protein you should target? So we take a mathematical modeling approach. So this is another model used in my lab that this is the artificial misfolded synuclein we generate. If you inject them into mice, you can see they can very efficiently induce this synuclein pathology or bad synuclein in the mouse brain. And if you look closely, you would see that these kind of pathological synuclein, they transmit in the mouse brain just as they did in the PD patient. And they transmit in a way not random, but very predictable based on the connections between neurons. So it seems like they are very willing to transmit between neurons that form connection with each other, but rather than just randomly go somewhere. That is interesting because number one, that indicates a transmission mechanism. Number two, that enable us to predict where they are going. So the whole mouse brain connection is known. Essentially, you have a map that tells you in the mouse brain which neuron goes to where. Because the synuclein transmission largely follows this map, we could use this map to build a mathematical model to predict where the synuclein will go. To make a very long story short, This is the quantification of pathology in over 200 brain regions to generate a detailed database. And we built this mathematical model to predict where the synuclein should transmit to. And if you compare the predicted value with this active value, you can play with the model a little bit to achieve a very high level of agreement between the two. You can see this is the best model that you can see the prediction is largely consistent with the active pathology. But you can also see that there are always some brain regions, like some of the dots is not on the line, right? That means these brain regions, they have a changed vulnerability for synuclein to develop pathology. So that actually gives us a window to see, okay, what is unique in these brain regions? What could be the genes or proteins here that is protecting us or make it worse? So with that idea behind this, we did a kind of incorporation of gene expression pattern into the model one by one to test the whole genome. And essentially, we built this very complicated gene regulatory network that based on the prediction should regulate the trafficking process that are very promising drug targets. And we even test them. You can see this is a gene that is called RORA. So if we overexpress this gene in the neurons, you can see with the expression of this gene, the amount of synuclein dramatically reduced. Right, the red signal is basically the pathological synuclein. So that is kind of shows the power of this mathematical modeling combined with experimental validation to identify potential drug targets to block this trafficking process. So the last strategy we are taking is targeting this site. So because if you think about this, for a bad guy to amplify the self, they have to convert the good normal synuclein into a bad synuclein. I was thinking, what if we have a modification or something for the good guy to protect themselves? Would that reduce the amplification of the bad guy? Actually, there is a lot of PTMs. So PTM is, you can think about it as a small tag that you can put on synuclein. So when you have this small tag on synuclein, it changes property. And maybe it will prevent this good guy to be converted by this bad guy. So we take it as a shield for the good guy. So to identify that potential shield, we perform like a very detailed analysis of all the possible tags on synuclein. You can think about this as all the potential different version of shield that we can give to this good guy. And then we test them, right? So this is testing of this kind of particular modification is called phosphorylation. And you can see we can test the phosphorylation at a different position of the normal synuclein and asking like which position will be protective, which position will be bad. And you can see this result. For example, this one is bad, right? Because if you give normal synuclein this kind of modification, the amount of pathology actually increased. That means this modification actually promote the formation of the bad synuclein. But look at here, if you give this synuclein this type of modification, that actually very efficiently reduce the synuclein pathology. So this is another version of the different type of modifications we tested. And also you can see we identified several that are very efficiently reduce the amount of synuclein pathology to block the amplification. So to summary, like the central goal of my lab is really fighting against this bad guy. So we do it in multiple different ways. And we target at the misfolding stage, we target at the transmission stage, and then we target at the amplification stage. So so we are very optimized that we should be able to get new therapies for PD. So this is I would like to thank my wonderful colleagues in the Movement Disorder Center, and this is my lab members. I like this picture not because you couldn't see our face clearly, but because you can see the light behind us, that is, you keep the hope, right? That you someday you will be able to find a new therapy for PD. And also, I want to thank my family. I always feel like doing science, like walking in water. You just have to keep going until you reach the target. Yeah, thank you so much.
Hey, thank you so much, Dr. Peng. Our next speaker here is going to be Dr. DiBiase, and she is going to tell us about this really interesting cell type called microglia that we've been hearing about this morning.
Good. All right, thank you, Will. It's really wonderful to see all of you here, and we're going to switch gears a little bit. You've been hearing a lot about what happens that affects neurons in the brain, and interesting. I was so glad that Will included the glial cells in his introduction, and Jeff has talked about them as well. So that's great. They're already getting a little bit of recognition. It's actually the case that only half of the cells in your brain are neurons. Half of the cells in your brain are non-neuronal cells or glial cells. And those are the cells that we study in my lab. And in particular, I'll be telling you about these particular non-neuronal cells, microglia. See if we can get the laser pointer going here. Okay. So where are these non-neuronal cells in your brain? Where are microglia in your brain? The answer is that they are everywhere in your brain. What you're looking at here is a cross section of a mouse brain. And this is a genetically modified mouse. So all of the microglia have been tagged with a fluorescent protein. So each of these little white dots is a microglial cell. This is a young adult, healthy mouse. And what I'm hoping you can appreciate is that they're all over. They're completely spread throughout the brain. So what this means is that all the neurons in your brain are completely surrounded by and interacting with microglia. So that's true for dopamine neurons. These are dopamine neurons in a mouse brain in the ventral tegmental area and the substantia nigra, pars compacta. We are all, of course, appreciating what important neurons these are. And they're very lovely. I love looking at this picture and seeing their amazing structures. But this picture is a little misleading because it kind of makes it look like they're just kind of there by themselves in the brain, which is not the case. They are completely surrounded by and permeated by microglia and also other non-neuronal cells. And what I hope this visually illustrates for you is that microglia and also other non-neuronal cells, they are really making the environment that the neurons live and operate in a really profound way. So let's talk a little bit more in detail about what is a microglial cell, what does a microglial cell do? They are sometimes referred to as the immune cells of the brain, and there are a number of things that they do that are kind of consistent with that identity. One of the things that they do is these branching cell processes of the microglia are motile. They're constantly scanning the brain tissue and looking for debris, also looking for bacteria or viruses that might have gotten into the brain. And if they find anything like debris or bacteria or viruses, they can engulf those things and remove them from the brain tissue. This is a process that we call phagocytosis. So yes, they do some important immune-related things in your brain, but that's not all they do. Over the last 10 to 15 years, we've learned that microglia are actually interacting with neurons all the time. So there are ways that microglia can help neurons form new synaptic connections. That's where information is stored. They also have ways that they can lead to removal of those synaptic connections between neurons. There are some ways that microglia can affect the electrical excitability of neurons. So this was really surprising to scientists in the field. These are, we thought of them as the immune cells, but yet we're learning that they're really impacting aspects of neuron function that are important for storing information and helping behaviors come about. Something else that's really noteworthy about microglia is that they are very sensitive to any sorts of challenges or insults that the brain might experience. So we like to think of them as kind of the first responders when there's some sort of challenge to the brain. So this could be an injury, could be an infection, or it could be the beginnings of a disease process. Microglia are going to sense that and respond to it. One of the things that they do when they're responding is something that Jeff mentioned. They can release inflammatory factors. Now, these are a mix of pro-inflammatory factors that are kind of increasing tissue inflammation that might be damaging, but there are also ways that they can release anti-inflammatory factors and maybe calm down inflammation. They also will interact with these toxic proteins. So we just heard a lot about alpha-synuclein and its transmission from cell to cell. Microglia are also capable of sensing these toxic proteins, and if they're in the extracellular environment, can engulf them and remove them from the tissue. So what I hope you can agree with is that if you think about all these different things that microglia can do, these cells are playing a really important role in kind of setting the backdrop for neuronal vulnerability and resilience throughout the lifespan. There are a lot of different ways that they can shape the health of the neuron. So I just want to show you a couple visual examples of some of those microglial functions that we talked about, because it's always nice to see visually. So I told you that the microglia are constantly surveying the brain tissue. So this is, do I have to stop the laser pointer to be able to play the video? And now, how do we turn it off? Oh, maybe just like turn it off. Like, oh, now I'm drawing. That's exciting. We're learning new things about PowerPoint all the time. Okay, it's off. Great. Okay, so these are microglia in the brain of a healthy young adult mouse. And you can see they're scanning the brain tissue all the time, looking for debris or pathogens that they might need to remove. And I think this is a really fascinating behavior of the cells. Now, I also told you they are first responders when there's some sort of insult to the brain. I'm going to show you an example with a very small tissue damage. So what you're going to see is if there's a tissue damage, microglia will sense it and immediately send their processes to that site of damage to kind of encapsulate it and then help with the repair process. So we'll play that one more time because that's one of my favorite videos. There's a little bit of damage. The microglia are like, oh, alert. You know, we're going to try to do something here. Okay. So I want to circle back to this idea that microglia are found everywhere in the brain. And I've just told you they can do a lot of important things to influence the health of neurons in the brain. I need to add one little wrinkle here, which is that even though microglia are found everywhere in the brain, they're not identical in different brain regions. There are some important differences in microglia that are found in different brain regions. And that's something that we study very actively in my lab. What is this going to mean for neuron health across the lifespan? Are there some brain regions where microglia are really good at helping the neurons stay healthy, and other brain regions where maybe they're not as good at helping the neurons stay healthy? And in particular, we're interested in what this may mean for Parkinson's disease. So I'm going to give you a little bit more information about these regional differences in microglia within what we refer to as the basal ganglia circuits. So these are circuits that are important for movement and reward-based learning, also other forms of learning. And it's also the circuits that dopamine neurons are part of. So what you're looking at here is tissue from a young adult healthy mouse and 4 different parts of these basal ganglia circuits. This is the nucleus accumbens, which is part of the striatum. And then we've got the ventral tegmental area and the substantia nigra pars compacta. That's where the dopamine neurons are, so that these guys get a star. And then the substantia nigra pars reticulata, which is another part of these circuits. And what I'm hoping you can see here is that there's an important difference across these regions in how many microglia there are. So the brain regions that have the dopamine neurons, they have fewer microglia than some of these other brain regions. We zoom in and look at their branching processes. There's also less branching of the microglia in the VTA and the SNC. And we've also looked at an important organelle inside the microglia that are called the lysosomes. This is, sometimes people will call this the garbage disposal of the cell. This is an organelle inside the cell that helps it break down and digest any material that it's taken up. And that's really important for cells like microglia that are taking stuff up from the extracellular environment. The microglia that are in the VTA and the SNC have fewer lysosomes than microglia in other brain regions. So I think this kind of, this supports that idea that maybe microglia in different brain regions are going to be more or less equipped to really support the health of neurons throughout the lifespan. How might that be relevant, particularly for thinking about Parkinson's disease? So we've already said that there are many ways that microglia can shape neuron health, and that the microglia that are in the brain regions with dopamine neurons seem different than microglia in other brain regions. It's also the case that many of these genes that we heard about that have been linked to Parkinson's disease risk, you saw the plot with many of the genes, and I'm just giving a couple examples here. Many of these genes are genes that are also turned on and have roles in microglia. So if there's a risk that comes from changes in these genes, part of that might be through what's changing about the way that microglia behave. We've also heard about environmental exposures and how that's an important thing that can increase your risk for PD. And we know that microglia are the first responders to various challenges. So microglia will certainly change their properties after things like mild traumatic brain injury, so concussions, repeated concussions. They will also change their properties after things like getting exposed to pesticides. And they remember those experiences. We refer to this sometimes as microglia becoming primed. So they can change their properties after these experiences and keep that change for quite a long time. And they may behave differently as additional challenges are encountered during the lifespan. And then finally, microglia change a lot as we are aging. And we've heard that aging is actually the biggest risk factor for Parkinson's disease. And this is one of the things that my lab has been most heavily studying, is how do microglia change during aging, and particularly within the brain region where dopamine neurons are located. Altogether, I think this really indicates that microglia have an important role in shaping the risk for Parkinson's disease. They are also equipped to shape the progression, how rapidly the disease is progressing. But there's also an opportunity here because these cells are really adaptable, and that makes them attractive therapeutic targets. And so we are a basic research lab. I'm not working with patients or human subjects, but we're hoping that we are going to be learning things that will show us how we could maybe harness microglia to prevent PD and slow the progression for individuals who are living with the disease. And I've got a great community of colleagues, many of whom are clinicians here, to be interacting with and try to see that we're translating that information towards therapies. Okay, so I want to talk more about microglia during aging and what we're finding. And you'll see various pictures of students and postdocs who are the ones in the lab pushing these experiments forward. I'm going to tell you about a couple different things that change a lot in microglia during aging. One of the things that changes a lot are their lysosomes. So we already talked about lysosomes, these organelles that are important for breaking down material that microglia have taken up. In a young adult mouse, the microglia have nice lysosomes all throughout their branching processes. In a late middle-aged mouse, so this is an 18-month-old mouse, which might be like a 60, 65-year-old human, there are some big changes. You see these really swollen lysosomes in the cell body, and we've really lost a lot of those lysosomes that are normally kind of scattered out in the cell processes. One of the things that's happening here is the microglia are accumulating these autofluorescent aggregates. These are basically damaged proteins and lipids that are filling up their lysosomes. So this is not alpha-synuclein. These are just a lot of additional proteins and lipids that are getting damaged during aging. But what's important to think about here is this is certainly going to affect how well the microglia might be able to deal with other things like alpha-synuclein that they might be taking up during the aging process. Now, we've been finding that these changes, the lysosome swelling and the formation of these aggregates, they are happening to microglia all throughout the brain, but they're worse in the VTA and the SNC compared to other brain regions. What we've been finding is that we don't think that these aggregates are benign because they're causing the microglia to proliferate, something I'm going to show you. And we also find that they're associated with changes in dopamine-dependent memory and cognitive flexibility. I'm not going to have time to show you a couple other things that we're also finding, but I'll just tell you about them. We're also finding that these changes and the accumulation of these aggregates is causing microglia to alter their expression of of many genes, but including genes that have been identified as relevant to PD. And it's definitely causing them to produce more inflammatory factors. So we see that local inflammation is increasing as this is happening in microglia. So let me just show you some images of the fact that this is causing microglia to proliferate and how does it relate to circuit function. This is just a picture of a microglial cell dividing. And you can see that these aggregates end up primarily in one of the daughter cells, and there's one daughter cell that isn't gunked up with this material. So maybe they're dividing to try to have some of the cells not be impacted by this material. And we've actually been able to collaborate through collaboration. There's a researcher, Dr. Carol Barnes at University of Arizona, who does a lot of work with aging macaques. These are monkeys. And we've gotten some tissue from her and seen that very similar things are happening in the VTA and the SNC of these aging macaques. You can see these aggregates mostly in one daughter cell, which is important because we want to make sure that the things that we're studying in mice are also happening in a primate, which is, much closer to a human. You can imagine as these cells are dividing, there's going to be more and more of them in the brain region, and that is something that we notice. So if you look at microglia in a couple different brain regions, from young adult mice all the way out to 2-year-old mice, this would be like an 80-year-old human, in that region of the striatum, there weren't very many changes in how many microglia you've got, but in the VTA and the SNC, where the dopamine neurons are, there are just these progressive increases in how many microglia are in this brain region during aging. And we think this is not something that's just benign, but that it really impacts the health of the dopamine neurons and the functioning of these dopamine circuits. The way that we looked at this was by doing some behavioral testing in young and aging mice. And what we do just briefly is we put a mouse in a very large box that we refer to as an arena. And there are some objects so the mouse can kind of orient itself. And there's a little bowl with sand and food treats. So the mouse can dig in there and find the food treats. So this is a reward-based learning. And then we take the mice out of the arena, and after either 2 hours or 24 hours, we put them back in the arena. And there's that same food well with the food treats, but 3 other identical food wells that don't have any treats. And we have to monitor the mouse's behavior and see if it remembers which one is the one that has the treats. So late middle age mice, again, this would be like a 60, 65-year-old human. They perform pretty similarly to young mice if there's a short delay, but they are not performing as well if there's a longer delay. And now we can look at these late middle age mice that are doing, these guys are doing pretty similarly to the young mice. They're performing pretty comparably. And these guys are the ones that are not performing as well. How does that relate to what's going on with their microglia? Well, in these late middle-aged mice, the mice that had more microglia in the VTA and the SNC had worse behavioral performance. So it seems like these changes in microglia during aging are quite important for how the circuits are functioning. Just to summarize that, we think that microglia with these overloaded lysosomes are doing a lot of bad things. They're proliferating, producing inflammation, and impairing the functioning of dopamine circuits. What we're doing now in the lab, and what I hope to be able to tell you about in the future, is we're working on ways to boost microglial lysosome function. And we hope that that can stop a lot of these processes and potentially protect dopamine neurons during aging. And also we're looking in mouse models of Parkinson's disease. I have like 1 minute to tell you just one other change in microglia during aging that we're really excited about. We've noticed that microglia start to contact dopamine neurons during aging physically. So this is an example of what this looks like, where you've got a microglia in green, and they're really hugging up against the dopamine neurons and their processes. Kiana, a grad student in the lab, has painstakingly reconstructed these contacts in 3 dimensions. And what has gotten us excited is that this microglia contacting the dopamine neurons increases during aging. And it seems to increase more in the VTA than it does in the SNC. And this is important because the VTA dopamine neurons are sort of more resilient to PD than the SNC dopamine neurons. So is this something that could be protective? And the SNC dopamine neurons are not getting as much of it. We also think this is protective because of how it relates to that same behavior I just told you about. This is sort of the reverse, where if you have aging mice with more contact between the microglia and the dopamine neurons, they do better on this behavioral task. So we think that microglia might be able to protect dopamine neurons by physically contacting them. And if there's insufficient contact for various reasons, that's going to leave the neurons vulnerable. What we're working on now in my lab is trying to identify the molecules that are involved in that contact. And so we can devise strategies to kind of enhance this microglial contact of the dopamine neurons and see if that's able to protect those neurons in mouse models of PD and potentially slow progression. So I'll thank the members of my lab, and you've seen we've got a great community of colleagues here, and that's I'm really grateful to be embedded in that community as well. Well, that's you. Okay.
So we've got about 15 minutes or so for questions. So I'm going to invite all of our first 3 speakers up here for questions. So if you guys maybe can stand over here. Give someone the microphone, you can pass it around. And I will come and give a microphone out to folks in the audience. So please raise your hand if you have any questions for our members. Want to start us off? Here, this is a microphone, so
Okay.
speak into that.
All right, there's a lot of discussion about the alpha-synucleins, and I'm not sure where they're headed because the alpha-synuclein biopsy that I had was for ruling in Lewy body. And I thought the presence of alpha-synucleins was relative to Lewy body, not Parkinson's.
So you had a skin biopsy?
Yeah,
Yeah, okay. So there's a skin biopsy called SYN1 biopsy. Take a little skin. It turns out that the nerve endings have synuclein. All the brain, all the nerves have synuclein in all our nerves, both in the skin, even in the brain. It turns out that Parkinson's, as I said, is not just in those dopamine neurons, it's everywhere. So if those dopamine neurons get abnormally, you heard Dr. Peng talk about PTMs, post-translational modifications. They add a phosphate group on it in a certain area. It's called phosphorylated synuclein. It's on one of the serines. And that is associated with Parkinson's. So that's the abnormal form of synuclein. So it's not a Lewy body, but it's telling you that you have Parkinson's because you have abnormal synuclein in your nerve cells in your skin.
And that can be positive both in Parkinson's and dementia with Lewy
Right.
bodies, which we call
Anytime you have abnormal synuclein, so there's other disorders that have abnormal synuclein as well. It's called multiple system atrophy, Lewy body disease, all alpha synuclein. All of those would be positive. So it's not specific to Parkinson's, but by far it's the most common.
other questions. I see somebody in the back there. Can you pass that over?
Hi, very briefly for Dr. Bronstein. You mentioned, am I on?
Yeah.
Okay. Environmental exposure to, you mentioned consistently pesticides, but are herbicides the same or different? Because I know like paraquat, for instance, is an herbicide, but it's associated with Parkinson's.
Yeah, so pesticides are herbicides, fungicides, insecticides, anything that kills pests. So all of those things are related. So it turns out, like I said, there's hundreds and hundreds of different chemicals, and only a few of them have been found to be associated with it. So there's certain herbicides, certain pesticides. So chlorpyrifos is only one. There's the ones you've heard probably the most about is paraquat. Big in the 70s, they used to spray it to kill all the pot plants. Rotenone is another one that's well known. And we found a couple others that Xyram, this whole class of fungicides, they tend to be particularly bad. So there's several different ones.
And for Dr. DeBiase, one of the slides showed, I guess, an earlier, younger mouse showed where there were fewer present in microglia in the regions where the dopamine neurons were. So simplistic, non-scientist me said, well, is there a possibility of introducing more into those areas so when they're still healthy, they could be doing more to prevent ultimately us getting Parkinson's?
Yeah, that's a great thought. We don't understand why in a healthy young animal are there fewer microglia in those brain regions to begin with. But that's a fantastic idea. Is there something about, as we are figuring out why there are fewer, can we mess with the signals that tell them how many of you should be in this brain region to just have more of them? I think that's a great experiment. It's a wonderful idea.
So raise several $1 million and we can do something about it.
Yeah.
Other questions? In front here? Oh, I'll come back to you. Right here?
Yes, I hear you guys talk about the inflammation, what it causes the inflammation in their body, and what we can do in the future to prevent that.
Yeah, I think that may be a question that many of us could comment on. We use the term inflammation, and as is true for many things, it's actually a broad term that covers many different inflammatory factors. There are many different specific inflammatory molecules that can increase in various circumstances, like if you have an infection. We see that certainly inflammation is increasing in the context of Parkinson's disease, and it's also increasing in the brain during aging. It's not unique to the brain. You will also see increases in what we refer to as systemic inflammation. So just what's going on in your circulating bloodstream and in other organs. There's definitely a lot of interest in how can we reduce inflammation. Some of the things that Dr. Bronstein mentioned about things, it's all the things that you've heard a million times, right? Like maintaining a healthy diet, maintaining some physical exercise. Sleep, maintaining healthy sleep is also something that can help with reducing inflammation. I think we're trying to also find additional ways to help reduce inflammation because not all individuals can engage in forms of physical exercise, or there may be individuals who have to have, due to their work expectations, can't maintain just like regular sleep schedules if they're involved in shift work. So we're also trying to find medicinal ways to help reduce inflammation. I'm not sure if that's a satisfying answer or if y'all want to add anything.
Yeah, just quick add. I mean, it is associated higher inflammation. And like I said, gut microbiome, the bacteria in your gut is associated with inflammation. That's why diet may be effective. But the truth is, we don't know. And there are experiments now going on, trials going on that are taking drugs that cut inflammation in the brain and in the body. And we're going to see if it slows it down or not. I mean, there's a small chance it may make it worse, right? They may be killing off good microglia. So we don't know, but we're going to find out in the coming years.
Great. We've got one question back here and then I'll go over there.
Yeah, Dr. Bronstein, you had a timeline slide fairly early in your presentation, like with the first symptom, like right in the middle of the timeline, and then it went back 20 years and went ahead 20 years. Was that mostly related to classic Parkinson's, or did it apply to early onset as well?
Yeah, so like I said, there's no Parkinson. You see one Parkinson patient, you've seen one Parkinson patient. Everybody's different. So that's just a gross generalization. So some forms, there's a form called tremor dominant Parkinson's that seems to be much slower. So there are different subtypes, and that's one of the things we just group of many of the people speaking, Dr. Peng, Dr. Zeiger. We're part of a big program that we just got a huge grant to study why some people progress faster than others, both genetically and environmentally. So it's a critical question. So that's just the average and just for representation.
Question over here.
Hi, this is a question for all three of you. It looks like Parkinson's is a very complex disorder. And so maybe one treatment doesn't solve it for all patients. How do you screen later on, or would there be a cocktail of different things to take for patients?
Yeah, I think that's that's an excellent question. It's like a precise medicine, actually, right? For example, I can kind of further elaborate that question. Like even for synuclein, different patients, different brain region may have different pathological synuclein structure as well. I think we want to start from somewhere. Like for example, the reason you target synuclein is because it's almost in every single PD patient. We hope that it could benefit as many patients as possible. But of course, there will be someone that may have a different structure or may have a other cause, like inflammation, maybe a major cause. I think back then, after that, there will be biomarkers to first identify each patient, and then that will provide guidance. Okay, maybe for this patient, for example, NERK2 mutation carriers or GBA carriers, maybe for them, like targeted therapy targeting NERK2 or GBA will be more beneficial for them.
Just to add on, I mean, I think that your question also included a little bit, would it be useful to be thinking about sort of combinatorial therapies and treatments? And I think absolutely yes. I hear the work that's being done with targeting alpha-synuclein and think, oh, wow, maybe there could be some good synergy between doing that. And if we can boost microglial methods of protecting dopamine neurons, I think there will hopefully be opportunities to be using multiple approaches to try to improve things for, as you said, the wide spectrum of specific disease phenotypes that we're seeing.
Just to add one layer, you really hit the nail on the head. So there's two aspects. One is how to identify each person, and then are there going to be specific treatments? So we're starting that. So for example, one of those big projects is we're going to be making stem cells with a collaborator at Cedars, Clive Svensson, from about 70 different people, and see if some people have different characteristics in others and whether that's the same as in people. So identify what caused your Parkinson's, what's not working well in you versus somebody else. So that's the identification, but we've already have some hints. So there's LRRK2 is the most common gene that's abnormal. Turns out about 30% have too much activity of LRRK2. So we're undergoing a trial that Dr. Thordarson is going to talk about a little bit later this morning, just for those people that have that variant of LRRK2. So that's an example of precision medicine. So just about 30% of people will be eligible, but it may work just for those people. So hopefully that's going to broaden to many different targets. And so each person might get their own treatment.
Our last question for the morning session right over here.
Hi there, thank you. Is it related age, aging is such a great risk factor for the compounds that you're trying to create, the AI assisted compounds that are being created. Is there a relationship between age and like therapeutic impact?
Yeah, that's kind of a good question. Actually, when we design that is really a structure-based design that is based on the synuclein structure in the patient. We haven't considered aging for that aspect, but maybe there is, for example, there could be a different synuclein structure for the patient who is really early onset versus those who's late onset, or in the patient that progress faster versus progress slower. So right now, we try to target as many as we can, but once we get more structured information for those potential diversity, we could certainly just shift the pipeline towards the particular structures.
Okay, so I'd like to bring up Dr. Cross now, and she is going to tell us about brain signals and walking in Parkinson's disease. Okay, take it away, Dr. Cross.
Can I move this aside a little bit, or
You can move it wherever you need to.
maybe I
I
can?
don't think that's better, unfortunately.
No, I thought it maybe would get smaller. This sounds echoey. Hello? Okay. Oh, still kind of loud. Who knows how to do this? Okay, can people hear me in the back here? We want to make sure everyone can hear. Yeah, better? Wow, still really echoey. Can you still hear me in the back? Yes? Okay, great. Well, thank you all for coming and sticking around after the break. I'm going to tell you a little bit, moving away from sort of the more basic science to studying patients and humans. So what can brain signals tell us about walking problems in Parkinson's disease? And I think the big goal is for us to get put out of business. So I'm going to try to address how we can improve treatments for people experiencing symptoms now. Hopefully the last 3 people and many others will make this unnecessary by curing Parkinson's altogether. But in the meantime, we still need to provide options for those of you who are suffering from walking problems and other issues. All right, so walking is a complex task, despite the fact that many of us do it very automatically and without thinking about it at all. We have to coordinate both sides of the body. The weight is constantly shifting over the planted foot to make sure that we maintain balance. We have to adjust our gait to all sorts of things in the environment, different obstacles, different goals, starting and stopping. And then we're also constantly taking in complex and dynamic scenarios around us, planning where to go next, predicting what might come out of the bike path. So to do this, walking requires coordination across multiple brain networks, as you can imagine. And in these deep structures here, we can think of this as the sort of autopilot for walking. These structures help to maintain the rhythmic stepping movements that occur without really thinking about it. But then they interact with many more superficial areas of the brain in the cortex to really adjust those walking patterns to the complex demands of the environment, to your own goals and plans and the context. Now you can see that the basal ganglia, which we've talked about a lot today, is right in the center of this coordination, coordinating these automatic gait walking structures with the more controlled contextual processes happening in the cortex of the brain. And so as you can imagine, if this coordination is disrupted, walking can be affected in many different ways in Parkinson's disease. So this just describes some of those observations that we have about how walking is affected. Even with straight walking, patients often describe this autopilot feeling like it's not working well, like you have to consciously think about every step you take. The result is that people walk more slowly, steps are shorter, they're shuffling. Steps are less consistent and rhythmic. There's variability in the length of strides and in the timing of steps. An indication of some balance problems comes from the fact that patients tend to spend more time on both feet with shorter swing phases of the gait. Starting walking can be difficult, so if you measure the shifting of weight, coordinating weight shift when you're taking that first step is different in patients with walking problems. Patients take many steps to turn as opposed to sort of turning on a dime like many of us do with intact walking. And then patients have more difficulty navigating obstacles or unexpected things in the pathway. So this is just to say that walking problems are extremely diverse. It's not just that you walk poorly or you walk well. There's many ways that walking is disrupted in Parkinson's disease. I think that thing's okay. And sometimes in some patients, walking just breaks down altogether. So in this phenomenon called freezing of gait, patients have difficulty even taking any effective steps, as you can see in this example here. People describe the feeling that their feet are suddenly glued to the floor. It can be unpredictable, very disabling and frustrating, obviously. And it really just is difficult to take that next step. And this is one of the more disabling features that can happen in Parkinson's disease. And unfortunately, our treatments are not always very effective for it. So this is a major interest in our research in my lab, is to try to improve and understand why this happens and use that to improve therapy for it. So obviously, we've been thinking about walking in Parkinson's for many years, and research to date has clearly demonstrated that there are brain differences across many regions in patients who have more walking problems. This is just from a review of many different studies that show that parts of the superficial cortex are affected, parts of the deeper structures that I mentioned, that autopilot. You can detect changes in the function, changes in the structure and anatomy of these regions. When you compare people with more walking problems or specific walking problems compared to patients who have fewer walking problems. And this is just to say, this is a complex thing, and many areas of the brain can contribute to poor function. So most of what was done in the past has been people lying down in MRI scanners or people seated. And that's what we've learned, that the whole brain really is affected in patients with walking problems. But this leaves obviously an important gap, which is how are these brain regions, how are these brain regions and these function in these complex networks related to specific walking problems? And how is the function in these very complex brain systems changing when a patient in that moment in time when the walking problem is happening? So in order to study that, the field has moved towards mobile brain imaging. In the last 5 to 10 years, it's been possible to record brain activity while patients are actually walking to really answer this question about what's happening in that moment when walking fails or when walking is disrupted. We can combine that with measurements of motion at the exact same time with very precise temporal precision. And this allows us to move forward and ask additional questions that we think are really important for understanding and treating walking problems. First of all, in situations and patients where walking is actually going pretty well, what does it look like? What is the goal? What do we want the brain to be doing when people are walking? And then an important next step is how does that activity change when walking breaks down, such as in freezing of gait? And then lastly, another important feature, I think, is how does the brain change when treatments work well for walking versus when they don't work as well? Because this can tell us what are those parts of the brain that are compensating, that are still intact and that can help us, that we might be able to harness as treatments. So these are sort of the three big picture questions that our lab is trying to answer with some of these mobile brain imaging techniques. Why does this matter? As I've already mentioned, we think that we can develop better treatments for walking problems if we can understand what's going on. And this is just one example of how we might use it. We already have ways to change brain activity in people. So many of you may be aware that in Parkinson's disease, we use deep brain stimulation to use electrical activity to change brain activity. And this can work really well for some symptoms like tremor and dyskinesias. But there's also a lot of other ways to change brain activity. And these are rapidly developing, and we're learning more and more about how we can change specific brain waves, specific regions, with things like ultrasound waves, magnetic fields, electrical stimulation provided from outside the brain. So we have this whole field that's telling us we can alter brain activity. But in order to use these to treat walking problems, we need to know how we want the brain activity to look, what are those signals that we need to change, and when do we need to change them, because many of these things can be perturbed in very precise time points. And this may sound a little familiar because we actually do have deep brain stimulation devices that were recently released that can record brain activity and then change the stimulation based on that brain activity to improve symptom control. Turns out these don't actually help walking yet any better than the original deep brain stimulation devices, and that's because we don't know how to change the brain yet to improve walking. So with our research, we hope to identify those signals that tell us there's a walking problem, and then use that information to change stimulation in some way to target specific signals that will improve those walking problems. In essence, we need to identify how the brain changes with walking problems so we can detect the problems when they happen and treat them with novel therapies. And to do that in real time while it's evolving. So I'm going to tell you about two projects that we're working on that get at these three questions I mentioned. First, I will tell you about one looking at what is changing in the brains when treatments are working. And here we focus on the effect of sensory cues. So you saw this video already of a gentleman having trouble walking, and down here he's also off medications. And you can see that when he steps over these visual lines on the floor, his walking is much smoother and more effective. He doesn't have those freezing episodes. And the same is true when you give a rhythmic sound. So these external stimuli can really help somebody walk in the moment. These are a well-described rehabilitation tool. But it turns out it's hard to use these in everyday life, and they don't really extend beyond this moment where they're being used, and people tend to habituate to them. So we can see that the brain is really walking better in the situation, but they don't work in real life. And so what we're trying to do with this is understand what is the brain doing when it's benefiting, and are those signals that are associated with benefits from these cues something that we could potentially augment to improve walking when it's not possible to provide those cues. So we do that in this study with a mobile EEG cap, like I showed in the diagram, and patients were wearing accelerometers on different areas of the body. We have a shoe laser that actually is available commercially that presents a line to help step over. And the setup looks like this. So you can see that these cues present this target to step over. We can record brain waves, electrical brain activity in real time and relate it to the timing of steps, and we can record acceleration of the foot and other body parts. And from those acceleration data, we can extract features of gait. How fast are people walking? How long are the strides? How rhythmic is the gait? And by putting all of that information together, we can ask what's going on in the brain when walking is improved with these cues compared to when people are walking with those lasers turned off. And not surprisingly, from much rehab literature, the sensory cues do improve walking in these patients. So here we see gait speed and stride length when people are walking with metronome cues in orange compared to the laser cues in green. And 0 is sort of the speed with no cues. And everybody improves, and older healthy adults also actually improve in their speed when you give them cues. And what you can see here that's also interesting is that the patient groups who reported not experiencing freezing of gait in their everyday life improved more than the patients who do experience freezing in both stride length and gait speed. So these cues are helping everybody, but they're not helping everybody the same. This is not new, but it is showing that the cues are working in our experimental manipulation. And what we're really interested in is what's happening in the brain while people are walking. So this is just a schematic to show you that we're seeing this dynamic brain activity. Here we're focused on beta activity. This is a signal when we measure it from the center superficial part of the brain that is a good index of voluntary movement control, of using those circuits that are important when we're making voluntary or conscious movements. And you can kind of see from here that we're getting some alternating activity. When the right leg swings, we have activity on this side. When the left leg swings, it kind of switches to the other side. So we have real-time brain activity here. We can quantify that and look specifically at sort of the voluntary motor control by looking in these central areas. And this is just another depiction in this beta activity that the left motor cortex and the right motor cortex are really alternating. And that's giving us good confidence that we're measuring brain activity, which is hard to do when people are walking, that is controlling voluntary movements. And we care about this because many people have suggested that cues help because they just help that voluntary system. The autopilot is broken and we use voluntary control to overcome it. But what we actually observe is that's only true for patients with freezing of gait. The group with freezing had more motor cortex activity with cues, and they also improved, although to a lesser degree than patients without freezing. And those without freezing don't show any of this extracortical activity. So this is important because it means people are benefiting from cues with in different ways. But it still leaves open the question of those patients who don't experience freezing have better improvement, and yet they're not using this same brain system. So we have some follow-up studies where we're looking at how these cues might be helping patients without freezing. And this really speaks to the fact that we may need to adjust treatments to specific patients and use different strategies. I'm going to race through the next one, although I like it. It's very exciting. Here we're looking at freezing of gait, looking at what the brain is doing when walking is good compared to when walking breaks down. We have a much more complicated setup here that I'll tell you about. Instead of just recording from the surface of the brain, we're also recording from deep in the brain with those DBS devices that I mentioned that can record brain signals and adjust stimulation. Here we combine it with motion capture, so we get a really complex view of what's happening during the movements. And here you can see a freezing episode. We can monitor where patients are looking while they're walking, and what patients are paying attention to has a big impact on their walking function, so that can give us additional information. And we're combining that with virtual reality in order to deal with a major problem in this field. So we want to record brain activity while people are having freezing episodes, as I mentioned. But what we've learned is that when you do that in a sterile lab with people paying attention to walking, they just don't freeze in the same ways. Freezing of gait happens because there's all this complexity, these different networks are interacting and disrupted, and you really need that complexity to reliably evoke those episodes and record activity. So we create these realistic scenarios that people describe as being triggers for freezing of gait. Using virtual reality, and we can actually show that if you increase the difficulty here on the x-axis, you can increase the amount of times people freeze, and that gives us a really powerful tool to look at how the brain is functioning when people freeze compared to the exact same scenario in VR when they're not freezing. A big question here is, can you really walk in VR with goggles on and not being able to see the environment? And so we did a whole study to make sure that you could. We have a real world, and we did a really high fidelity recreation of that in virtual reality. And this is just an example of one person walking. You can see here there's nothing there, but he's seeing that in virtual reality. And he has freezing episodes. And if you look here, he's walking quite similarly when he's wearing the VR goggles and when he's not. And as he approaches the obstacle again on the way back, you'll see that his freezing is also quite similar. He has a little episode, he hesitates, he has another hesitation, and you see that also here with the real obstacles. So this, we quantified it in a lot of patients, and we just showed that in orange and green VR in real life, there's really a high fidelity of what situations cause different patients to freeze. If they don't freeze in VR, and if they do freeze, they freeze in the same situation. So now we know we can use this to provoke freezing. So this has set the stage for our current study that we're starting up now, 5 years funded by the NIH, to really combine all of these tools to elicit freezing episodes, record brain activity and motion capture activity, and then we can say right before the freezing episode, what is happening in the brain? How did that change from when they were walking well? And then how did they overcome that episode? Is there some way we can help people get through the episode and initiate gait faster or easier? We can compare these brain activity in people who freeze and people who don't freeze to see if there's something that's causing the susceptibility to freezing. And so with this platform, I think we have a really powerful tool to ask many, many questions with regard to what's the brain doing during gait. And hopefully I've convinced you that by doing this, we'll be able to identify new ways to improve walking with adaptive stimulation therapies and other tools that are emerging. With that, I want to thank first and foremost the patient volunteers. None of this would be possible without a lot of effort and time and probably discomfort sometimes from the hours that people spend with us, some people even here in this room. Obviously the funding support and then my lab, and most of this work is really spearheaded by Leo Ma, who's a postdoctoral fellow, physical therapy doctor, and also a PhD. So thank you, and forward for questions.
All right, so we'll save questions for the end again, and now we're going to turn it over to Dr. Galbatan.
So, preclinical development of drugs. What is that? We all know about clinical trials. This is where we're going to get the drugs, hopefully one day, to the clinic. But before that, there is a lot of work that is done in the laboratory using test tube tests, cell culture, animal models. And there's some similarity to the clinical trials, but some interesting differences. So when we start such a project in the lab, the first thing we want to do is we want to test, is our treatment, is our molecule, is our antibody going to have efficacy? Is it going to hit the target we want to hit, and is it going to have a beneficial effect? We test, as Dr. Peng told you, a lot of molecules, and eventually, if we find a good molecule, the next thing we do is we want to make sure it has low toxicity. We want it to be safe. If that works, we also test something called pharmacokinetics. Pharmacokinetics is how the drug, the treatment, interacts with the body. Do we need to give it as an injection as opposed to an oral drug? Is there some other route of administration? How long does it stay in the blood? Does it get into the brain? How fast does it get cleared out of the body? All of those are really, really important questions that most people don't think about. You just take a drug, you assume it's going to work. But all of these we have to answer before something can become a drug. When we get, hopefully, out of many, many thousands of molecules that we screen, to one that looks really promising and can get into a clinical trial, then the order is a little different. And this is based on what the FDA requires, because the first principle is do no harm. So the first thing we test is safety. And only if it's safe in humans can we move on. Typically, the next step will be to test the pharmacokinetics, make sure that it is in the body and reaches in the body where we want it to be. And then we test, does it actually does it achieve the effect we want it to achieve? So if you talk with people in the pharmaceutical industry, they will tell you until they're blue in the face about all of those clinical trial stages, phase 1, phase 2, phase 3, etc. But a lot of the work that has been done in the lab, in the preclinical stage, they just call it phase 0. This is what we are doing here at the university, and it takes many years to get to the point that we can get a drug into clinical trials. Well, in my lab, we have been developing these molecules that we call fondly molecular tweezers, even though it's really a misnomer. They don't tweeze anything. They just look a little bit, actually a derivative. The first derivative looked more like a tweezer. These don't look like a tweezer. I think they look more like a horseshoe. But the name stuck. And they were discovered about 20 years ago when I still didn't know much about Parkinson's disease. I was more of an Alzheimer's researcher. But similar to Parkinson's, in Alzheimer's disease, there is this problem of protein clumping. And so I was looking for molecules that could prevent or disrupt the protein clumping and found these molecular tweezers coming out of an academic lab in Essen, Germany. I read the paper, contacted the professor there, and he was gracious enough to send me some of the compound. We started testing it here so that it did what we hoped it would do. And pretty soon we discovered it wasn't just good for the proteins involved in Alzheimer's disease, but it had a broad spectrum activity. And in the context of Parkinson's, we thought, okay, let's test it against alpha-synuclein. We know it clumps together. We know it becomes toxic. Can it work? Turns out it can. So this is one of the common tests we do in the lab. How do we know that the protein clumps? We start with the normal protein. We add a specific dye. It's called thioflavin T that has this characteristic that if a protein aggregates near it, it will bind to it and it will start emitting fluorescent light. That's very useful to us. We can measure this fluorescent light. We know the protein aggregates. As you can see on the x-axis here, we're looking at hours. So for hours, for a couple of days, nothing happens. We're shaking the protein, and then all of a sudden it starts to go up. And if we're just using the alpha-synuclein on its own, this is the red line, it will get high until all the normal protein has consumed, everything has transformed into the amyloid fibrils, and it plateaus, no longer increases. When we added the molecular tweezers, now we saw less and less of the aggregation. So it took much longer until we started to see the fluorescent light, and the final level was lower. We then looked at the protein under the electron microscope, and we saw that indeed, when you don't add the molecule, it forms these amyloid fibrils. When you do add the molecule, it still forms some clumps, but they don't look the same straight fiber-like structures. They form what we call amorphous aggregates. So the next question is, well, are these amorphous aggregates that form with the molecule going to be still toxic or not? For that, we use cell culture tests. These pictures are not from my lab. We didn't have good enough pictures, so I looked on the internet and found something that looked very similar. So the top picture is cells that are healthy, happy. That you see that they have this rhomboid shape and they have some lines. These we call processes or neurites. They are akin to the axons and the dendrites of neurons in the brain. Once you attack them with something that is toxic, you see how they fall apart. The cell bodies become kind of these little round things and the processes start to break down. We added alpha-synuclein to our cells and we measured percent viability. And you see that when we add no molecule, we get about 30% reduction in cell viability. So it starts at 70%, and then we add little by little increased concentrations of the molecule until we can completely prevent this toxicity. So that was a good sign. Indeed, the clumps that are not fibrils are also not toxic. Then, as an Alzheimer's researcher, I started participating in the group meetings of the Parkinson's group here. And the first time I presented it, Dr. Bronstein, I remember still, it was, even though it was 15 years ago, said, I have an animal model that can give you an answer within 2 weeks. Of course, my mind was blown. We started collaborating. Dr. Bronstein added our molecule into the water in which his little zebrafish embryos developed. And as you can see, if you untreat them, but you transfect into them the toxic alpha-synuclein, what develops doesn't look like a fish. It looks like an unfortunate clump that dies very quickly. But when the molecule was added at 2 different concentrations, all of a sudden they did look much healthier. And this is a pretty complex graph in which Dr. Bronstein and his group quantified all of these data, but green is good, red is bad. And you can see that with the increased concentration, 1 or 10 of the molecule, we get more green. And that over time, most of the red lines decline because these embryos are dying, but the green ones remain healthy. That was obviously very encouraging. And with that, we said, okay, this is a great model. Now, what about something that looks a little more like a person than a fish? Obviously, that's the mouse. We all know that humans are just big mice, right? So for that, we collaborated with another group, Marie-Françoise Chesselet, who retired a few years ago, no longer at UCLA, but she had a good mouse model of Parkinson's disease. These mice express the human type of alpha-synuclein throughout their brain, and they develop motor deficits. We can measure them, for example, with the rod test. You put a mouse facing up, and it has to turn and go down, or with the challenge beam. This is this mesh, wire mesh. You can measure how many errors per step the mice are making. So we treated these mice by an injection, and this injection is not, even though it's a daily dose, it's not everyday injection. It is a special pump filled with the drug that you can implant under the skin. It releases the drug into the bloodstream on a constant basis. You can see that the dose of the drug, which I'm going to return to, is pretty low. It's less than 1 milligram per kilogram. And we did that for a month. So I'm just going to show you one result out of this study. On the left is normal mice, and you can see that they make very, very few errors per step, and the drug doesn't do anything for them. The Parkinson's mice make a lot more of those errors in the white squares, and the drug substantially reduces the number of errors. Now, because humans are not really big mice, one of the issues we face when we do preclinical development is that no one mouse model really recapitulates all the aspects of the human disease. To address that, typically what we will do is we will use a number of different models. Each one is a little different, and if we get consistent results in these models, then we have more confidence that our drug may be useful for humans. So here's another example of a mouse model that has the alpha-synuclein. I don't know why the other ones are, oh, now they're moving, okay. Here, what we're measuring is a psychological aspect of the disease. Mice, just like us, when they feel sick, they want to sit at the corner, stay close to the wall. They feel more protected there. When they feel healthy and happy, they will go much more into the center of the arena. So we're measuring basically the number of entries and the amount of time that they spend in the center versus the periphery of this arena, and we call it an anxiety-like phenotype or anxiety-like behavior. And then when we quantify that, we can see that the healthy mice have a certain ratio in which that they spend in the center versus the periphery. And those that receive the vehicle, so no drug, but these are sick mice, much lower. And then when we add the drug, we kind of normalize their behavior. Here are two other models. The first one, the top one, is another genetic model where a number of behavioral tests show that the normal mice, what we call wild type, behave in a certain way. All of the transgenic ones that are marked here as OVX have a decreased ability to perform whatever the task is, and with the drug, they get back or close to back to normal. On the bottom is one of the models in which Lewy bodies from a patient brain is injected into the brain of the mouse, and then we measure how many of the dopaminergic neurons survive that. So without drug, not many of them, and you can see a lot less brown on the left. With the drug, we preserve those dopaminergic neurons and they remain alive. So quite a lot of what we call good efficacy data. What about the safety? Well, we did a safety experiment here at UCLA with the help of the veterinarians. We started with a large dose, 100 to 1,000 times higher than the one that was needed for good efficacy. And we found, as you might expect, all drugs are processed by the liver. The liver did see an injury when we had such a high drug dose, but all the other organs were perfectly protected. We didn't see any problems there. Then we did a chronic treatment for a month with a lower dose, still much higher than the dose that was required for the efficacy. And there, what we saw was one side effect, which was reduced cholesterol. So if you have to have a side effect, that's not the worst one to have. Pharmacokinetics. We want to know that it interacts with the body the way we hope it would. So we saw that it remains in the blood for about 2 to 3 hours, a little shorter than what we hope. And what we also found was that we could not give it orally. When we gave it orally, it did not cross from the gut to the blood. That was unfortunate. We did find that by injection, by this injection under the skin, as opposed to injection into the vein, which is what chemotherapy therapy is, subcutaneous injection worked well. And it even increased a little bit the penetration into the brain. So the drug does get into the brain. And what is fortunate about it is that as opposed to the clearance from the blood, which is very fast, in the brain it remains much longer. So we can give a very low dose, it gets into the brain, it accumulates there to what we call a steady state and achieves the therapeutic effect. I do want to take a few minutes to explain this because this is important. We want to understand the mechanism of how this works in the brain. So here I have to diverge a little bit into what Dr. DiBiase talked about earlier, which is lysosomes. Lysosomes are these little compartments inside the cell where proteins get degraded. They get degraded by a combination of acid and enzymes. And this is where the cells will send the alpha-synuclein that is aggregated to get rid of it. But there's a problem. The moment alpha-synuclein goes from the normal pH that is not acidic in the cell into the lysosomes where the pH is acidic, it wants to aggregate much, much, much faster, orders of magnitude faster. So the degradation, the breakdown of the alpha-synuclein has to be really fast and really effective. And Dr. Degas showed us that it becomes less and less effective with age, and there are genetic factors and environmental factors that Dr. Bronstein told us about that can lower the lysosome activity. Well, lo and behold, what we found is that our drug likes to concentrate exactly there in the lysosome. So it gets into the cell. Some of it remains in the cell in general, but a lot of it goes into the lysosomes, which is exactly where it is most needed, because this is where the cells send the aggregated alpha-synuclein, and this is where it can help prevent the aggregation or inhibit the proliferation of the aggregates. So we showed that we have efficacy. We showed that the drug is safe. The pharmacokinetics is not exactly where we want it, but it's a good start. We can improve it. We have a good idea about the mechanism, and that led last summer to the creation of a new startup company that I created with a few other founders. And I can tell you that we have completed something called technology transfer that is really important. Our CEO told me that she had to close down her last company because the technology they adopted out of Harvard just did not reproduce out of the labs of those researchers. So we were all biting our nails, but they confirmed that everything out of 4 different labs has now reproduced well by the company. We completed a synthesis of the compound, which is quite complex at 20 times higher scale than what we could do in the lab. We completed some safety screens. Pharmacology in rats and dogs looks similar or even better than in the mice. And we even got some clues about the mechanism of how the compound gets into the brain. So hopefully, if all goes well, next year we will get to the point that we can start recruiting patients for a clinical trial. I just want to thank my collaborators, especially Dr. Bronstein, who was so instrumental in getting this off the ground, Marie-Françoise Chestelet, who helped with the mouse experiments, and all the former and current members of my lab contributing to this project, including Dr. Kara Schreiber, who's sitting here in the audience and has come here on a Saturday morning out of her break to be here in this exciting conference, and thank you.
All right, our next speaker is Dr. Thorderson, who is going to give us an update on clinical trials that are happening here at UCLA. I'll let you take care of that.
Thanks. Okay. Can you hear me okay? Great.
There you go.
Awesome. All right.
Stick around. We'll have questions at the end of this presentation, and then we have some more food out in the lobby to enjoy after that. So thank you very much.
So hello, thank you all for hanging in here and sticking it out to the very end with me. My name is Danny Thorderson. I'm one of the movement disorder neurologists here at UCLA, and I have the privilege of talking about our current clinical trials program at UCLA. So there is a huge unmet need in Parkinson's care. So what is great is that we have great symptomatic treatments for Parkinson's that work quite well to alleviate these symptoms. But unfortunately, often as disease progresses, some symptoms of Parkinson's may become more difficult to treat with these treatments. And right now, we do not have a therapy that slows or stops the disease from progressing. So I'm going to talk today about a few ways in which our clinical trials program is attempting to close this gap through a few different approaches. So what is a clinical trial? We have to, before approving a treatment, really thoroughly assess whether a treatment is safe and effective. So every intervention, be that a drug or a surgery or a device, has to go through several phases of clinical development before it can be approved for treatment. The first phase is phase 1, which is really aimed at looking at safety. So this is the first time that a drug is tested in humans. It looks at it in a small number of participants and essentially just assesses, is it safe, or are there problems that this drug could potentially cause in someone's body? I want to just emphasize also, hopefully you've gotten a sense of this from our talks today, there is so much work that happens before we can even get to phase 1, right? So, and then in phase 1, if drugs are determined to be safe, we can move on to phase 2, where we continue to look at safety and make sure that there aren't problems coming up with this intervention. And then we begin to look at if the treatment is helping or not. In phase 3, we look at this intervention in larger groups of patients. And this is the final phase where we really aim to definitively assess if a treatment actually works. And this is generally the final step before approval. You can think about treatments in 2 broad categories. Symptomatic treatments improve the symptoms of a condition or alleviate various symptoms, be they motor or non-motor. And in disease-modifying treatments, we actually attempt to slow or stop disease progression by addressing the underlying pathological processes and preventing neurodegeneration in the case of Parkinson's disease. So I'll mostly focus on disease-modifying therapy approaches today. I won't go too much into this because I think our other speakers have really already outlined what we believe to cause Parkinson's disease, but we know in the vast majority of Parkinson's patients, we see these alpha-synuclein clusters or aggregates that then unfortunately can spread from cell to cell and cause progression in Parkinson's disease. So we have a lot of evidence that this is a key pathological factor in Parkinson's. So there are a number of different ways that we can try to target, either this toxic protein aggregate or other mechanisms that we can try to help cells compensate in stressful situations. So you can think about these as either upstream kind of precision approaches that might target specific organelles such as mitochondria, or you've heard a lot about lysosomes today. Other approaches that might target specific pathways like the LRRK2 pathway, which I will talk about, or more broad approaches that either target alpha-synuclein, maybe preventing it from aggregating, for example, or preventing it from spreading from one cell to the next. So I'm going to talk about one of these broader approaches first, which are anti-alpha-synuclein antibodies that aim to prevent spread from cell to cell between neurons. So there have been 2 major drugs, anti-alpha-synuclein antibodies that have been tested in Parkinson's disease so far. The first one was cinpanemab, which was Biogen's drug that unfortunately did not meet its primary endpoints and was abandoned back in 2021. And then the second one was prasinezumab, which has been studied in a couple of clinical trials so far. The first one in which it was studied was the Pasadena trial. So if you came to the Levine symposium a few years ago, you might recognize this slide. The Pasadena trial randomized patients to either receive this drug, prasinezumab, or placebo. You can see in the blue here that these are the patients who did not receive prasinezumab. So higher up on this graph indicates more symptoms or a higher burden of symptoms or worse Parkinson's disease. And you can see at the end of this 1-year treatment period, unfortunately, there was not a difference between the groups that were treated with prasinezumab or the group that did not receive the drug. But the other thing that you can see is that these curves do look like they separate. So perhaps maybe there's a slight signal of efficacy that we just couldn't quite detect. The Parkinson's disease, as we know, is a slow disease. So perhaps 1 year was not enough time for us to really assess if this drug is having a benefit or not. So what happened at the end of the Pasadena trial is they extended this trial into what's called an open label extension. So that means that everyone in the treatment group and everyone in the placebo group was given the option to go on treatment or receive the drug. So now the problem with open label extensions is that we don't have anyone to compare them to now. We can't really say, is this progression different from the people who might not have been on the drug? So what the investigators did in this paper is they looked at an external comparator group. So they took a group of patients from the PPMI study, which is the Parkinson's Progression Markers Initiative study. And they basically thought of this group as representing an average Parkinson patient or average Parkinson's progression over time. They continued to follow this group and they followed the prasinezumab treated groups from the Pasadena trial. And what they saw was over a longer treatment period, over 4 years, perhaps there was some stabilization of these patients' disease progression over time. So we can't really say anything definitively because that external group could be inherently different from our treatment group in some way. But based on this, and then based on the results of another phase 2 trial called the Padova trial, which narrowly did not meet its primary endpoint either, they did decide to continue with clinical development of this drug. So when they looked at certain subgroups of patients within the study, they did see that perhaps there was a signal of benefit. So based on this, the study sponsor, which is Roche, has announced a phase 3 trial, which is called the Paraíso trial, which we're really excited to be a part of here at UCLA. This is a global trial, so that means we're testing it in many, many patients all across the world. And they intend to enroll people who are in early stages of Parkinson's disease who are on levodopa treatment already. So that's one of the kind of key differences between this trial and the prior trials. The intervention will be the same. It's the prasinezumab, which is administered IV versus placebo. And this time, importantly, they're studying it over a much longer treatment period. So this trial will be at least 2 years of a double-blind treatment period to really attempt to definitively assess whether this drug is preventing progression in Parkinson's disease. So we're extremely excited about this study. I'm going to switch gears a little bit because I think the PD generation study is an important segue into the next trial that I'll talk about. The PD generation study offers free genetic testing to patients with Parkinson's. It looks for the seven of the most common genetic mutations associated with Parkinson's disease. It includes free genetic counseling, so someone will go over your results with you, explain what a positive or a negative result might mean. And we are a super site at UCLA, meaning that we offer participation here in Westwood as a one-time visit, or you can even do it from home. We can send a home blood draw kit to you with support virtually from our study staff. So why do the genetics of Parkinson's? Well, I'm probably now sounding like I'm parroting a lot of things that people have already said today. But by looking at the genetics of Parkinson's disease, we can figure out what might cause Parkinson's disease in a given person. We know that no two people with Parkinson's look exactly the same. And what we found is that probably different underlying processes are causing Parkinson's in different people. So there's a lot of interplay between these different processes. But ultimately, it kind of culminates in the same final problem, which is loss of these dopamine neurons in the substantia nigra. So you can kind of think of Parkinson's not as just one disease, but as a number of different processes that all end with the same problem. So this is really the future of the field, and we've kind of already touched on this a little bit in some of our Q&A. What causes Parkinson's in Joe might be different than what causes it in Sally, and there may not just be one answer for slowing disease progression in a single person. It probably will take a combination of different approaches in order to slow disease progression in a given person, and it may take a different combination of approaches in the next person to actually slow progression. So it's a complicated problem with a complicated answer, but the future of the field is really to try to tailor these therapies to every individual. So one example of a precision medicine approach in Parkinson's disease is in LRRK2 associated Parkinson's disease. So the G2019S mutation in LRRK2 is one of the most common mutations that's seen in Parkinson's, and it results in increased LRRK2 kinase activity. So there's a hyperactivation or overactivation in this pathway that causes a number of downstream problems that results in neurodegeneration and enhanced spread of alpha-synuclein. And we have seen now in preclinical studies, there have been drugs that have been developed to turn down the activity in this pathway and try to normalize the activity in this pathway. It's been studied in animals, and early testing for safety has been conducted in humans, and now there are a number of phase 2 trials that are being conducted in kinase inhibitors. And we are extremely excited to be a site for one of these trials called the NEWLARK trial. Again, this is a phase 2 trial of an oral medication that inhibits that kinase activity. In order to qualify for this trial, they are trying to enrich the population. So they're really trying to look for people who actually have high activity in this pathway. And for this trial, they've developed an investigational diagnostic test that looks at a number of different genetic markers to really determine if a person's disease might actually be driven by this LRRK2 pathway. So it's really interesting because you don't necessarily have to have a LRRK2 mutation in order to qualify for this study. So really anyone with very early stage Parkinson's disease is encouraged to engage and possibly enroll in this study. They are looking for levodopa naive patients, so people not yet on carbidopa levodopa, which I realize eliminates a lot of people, but the eligibility then is determined through a saliva test that you just spit in a little tube, mail it off, and then find out about a month later whether you might qualify for this trial. All right, I'm going to switch gears a little bit and talk about symptomatic and restorative therapies. So stem cells in Parkinson's are always a hot topic. This allows study of Parkinson's in a number of different ways. Dr. Bronstein mentioned looking at different stem cells from different patients to try to examine why Parkinson's is happening in you versus you versus you. And then the other way in which stem cells are being used in Parkinson's is through transplantation to replace the lost dopamine neurons, implant cells that differentiate into these neurons and reintegrate and reinnervate the striatum, which is these kind of circuits in the brain that help facilitate movement. So this is a really exciting approach. This entails surgical transplantation of stem cells into the deep structures of the brain, and then immunosuppressive regimen to help promote the survival of those cells. This is not the first time that we're studying stem cells in Parkinson's. This actually was done quite a while ago. And in those early studies in the early 2000s, they did not meet their primary endpoints. There were some patients that developed some problems from the stem cell grafts. But when they looked at these patients over a longer period of time, they actually saw that many of them did quite well and did not have to be as dependent on medications in the long term. So based on this information and also based on really exciting advancements in stem cell technology that have come out in recent years, there's been kind of a reinvigorated interest in this area. So Blue Rock Therapeutics has developed a stem cell product. They have a really, really sophisticated way of taking a cell population and proliferating it so we don't have to worry about where the stem cells come from, and then really steering it in a very precise way to differentiate into the exact type of dopamine neuron that we need in those deep structures of the brain. So they have this cell product called bemdaneprocel. It was studied in a phase 1 trial a few years ago. So 12 Parkinson's patients received a stem cell transplant. And it did meet its primary outcome of safety and tolerability at 18 months. So they, based on this, have announced a phase 2 trial, which we're extremely excited to be a part of here at UCLA as well. It's called the Expedite 2 trial, where we are now continuing to assess safety and also beginning to assess whether this might be effective in reinnervating these motor circuits and helping with the motor function of Parkinson's disease in particular in the long term. So this study is looking for patients who have had Parkinson's disease for several years already, and those who have good benefit from medications, but perhaps over time may not have sustained benefit from medications throughout the day, so leading to hours of what we call off time, or times that medications are not working well in one's system. And there will be a control group in this study, and patients will be given an immunosuppressive regimen to help promote those cells' survival. I wanted to just also kind of briefly touch on some common concerns that I get or questions from patients when it comes to clinical trial participation. One being, will I get placebo? This is a very real concern. Some patients do get placebo in many of these trials. Many of them are designed, however, so that more patients get the active drug treatment. You have a higher chance of getting active drug than you do of being in a placebo group. And then sometimes patients, at the end of the trial, there's the possibility that the trial may extend into what we call an open label extension, where everybody who was involved in the trial is offered that treatment at the very end. We make it very clear up front what's the probability of getting placebo, and make sure that you understand what you're getting yourself into when you're enrolling in a trial. Is it safe? These studies that I mentioned today are all in phase 2 or phase 3 of development, so they already have gone through early safety testing. We have really thorough processes to ensure that trials are done ethically and that we are protecting human subjects. And then, is it a big time commitment? So that depends on the type of study that you might be enrolled in. It could be anything from, an observational study where you fill out a survey to a really in-depth clinical intervention where you have to come back. But we do our best to make participation as easy as possible for you. How to get involved? Start by talking to your neurologist. They know you the best and your Parkinson's the best, and they would probably have the best sense of what type of clinical trial might be the best fit for you. You can join registries and let us know that you're interested. And I'll just say, even if a study isn't right for you now, there may be one in the future that may be better suited to fit your specific needs. I'll also just say that this is an incredibly exciting time in the world of Parkinson's research. There are so many treatments in the pipeline, reaching phase 2 and phase 3, kind of getting closer and closer to that finish line of approval. There are so many intelligent and motivated people working on this problem. So it's really an exciting time, and progress doesn't happen without your participation. So thank you all so much for being here to just engage with our group today and hear about what's going on at UCLA. And I will stop there, and I think we're going to do a Q&A now.
Let's bring up Dr. Pross and Dr. Baton, and we'll take questions for a few minutes. Hey, who has questions?
Yeah, for Dr. Cross, the technology that that you showed with the lasers and all of that that was so effective, is that available?
Yeah, so those lasers are available. There's a bunch of startups and small companies creating queuing devices like that. That one is called Walk with Path. If you Google that, you'll probably find it. It's exorbitantly expensive, about $1,000 for the consumer. The problem with a lot of these things is that they are marketed outside of sort of, marketed direct to consumer, and so not covered by insurance. And
Did you say the name was?
Walk with Path. It's an English company somewhere in Europe. And there are various sensor tactile ones and auditory ones. Many patients find, as I described, that they sort of help in the moment, but they are hard to implement in real life. And so I think that's also something I would caution if you're going to spend a lot of money, that there's varying experiences with it.
Other questions?
My father very recently had an adaptive DBS device implanted. He just had his final surgery in February. How much do you think, like Dr. Cross, you were referencing how you want to optimize the adaptive DBS? That I've never heard anything like that before. So how long into the future do you think that some adjustments will be made for, fine tuning the adaptability of these DBS devices?
That's a really good question. So far, what we know about adaptive DBS is that it works as well as non-adaptive DBS. We implant lots of patients without adaptive DBS still because there's not enough data and experience, as I sort of described, to know how to use it to actually get a better benefit than non-adaptive DBS. In the clinical trials that were run by the device company, many patients chose the adaptive program over the non-adaptive. So some people sort of felt better. But if you looked at the objective measures as a group, there wasn't an overall benefit. So, I think many of us who have tried using it, if there's room for improvement over regular non-adaptive DBS, we try it. And we have to assess on an individual patient basis if it actually helped. And in my experience, sometimes it is better and sometimes it's not. And so that's exactly as you described. We don't know how to use it yet. So that's really something that we're going to get with experience now that it's out. Many places are, trying to do registries and things like that. The device company is trying to come up with ways to help clinicians know how to use it in a way that's effective. So I would say on the order of years, how we really know who it's going to help and how to apply it.
That's really interesting. Thank you.
Any other questions over here?
Thank you. This is for Dr. Batan. You're entering phase 1 experiment, right? Clinical trial. Is it going to take place at UCLA? And ideally, if everything goes well up until phase 3, when do you estimate that you'll have a drug in the marketplace?
Wow, I wish I had a crystal ball. So once the company was established, a lot of things got out of my hands, and now they are dictated by the people who run the company. I still am in an advisory capacity. So I talk to them, I tell them what I think, but it's their decision. Like I mentioned, I hope we can get to the beginning of a phase 1 in 2027. Depending on how that goes, I think there is a chance of partnering with pharmaceutical companies, which can accelerate things, but I would hesitate to give you a real timeline.
All right, other questions?
How is a mouse with Parkinson's developed?
That's a question I think many of us could answer. So there are many ways to do that. The first is probably genetic manipulation. You can insert a gene from the human genome into a mouse that would cause some of the causes that we know lead to Parkinson's disease. There are now a lot of models where you take the alpha-synuclein fibrils. Dr. Pang has shown some of that, and you can inject them directly into the brain to cause something that is that resembles Parkinson's disease.
Maybe we'll take one last question.
So I understand that we're talking about crystal balls, but between a chemical approach and a genetic approach, I guess stem cells you say are in phase 2. Those of us who are with Parkinson's, is this going to happen in our lifetime or?
I strongly believe it will happen in the next 5 to 10 years. That's me, just guessing, betting, right? I think, it's going to be, a number, as I mentioned, a number of different approaches. So while we may have something that begins to slow progression slightly in the next several years, really to stop Parkinson's in its track is going to take a combination of different things, right? But I am very, very hopeful and optimistic that we will have something very soon.